Right. Hey everyone, my name is Lucas Liu, and I'm a postdoc researcher in at Sony's lab. So I had my training in computer science, and now my research focuses on building AI models for cancer research and precision oncology. And my talk is going to be a little bit different from all the awesome speakers' talks before. They focus on understanding biology and data sharing, all that stuff. But my presentation is gonna focus on the predictions because of my restricted computer science mind. But one thing I'm gonna do similar to all the speakers is I'm gonna cut my presentation short into just 8 minutes. So my talk is going to focus on how do we use AI and stats together to predict or trying to learn the molecular alterations or biomarkers from H&E pathology images, especially focused on the prostate cancer. Just a little bit background of the clinical motivations of this project. We're trying to the molecular alterations are really critical for in cancer care. So especially for prostate cancer, which is what our project focused, we're trying to, one of the key biomarkers called MSI high. So if we know that if the patient is MSI high, that we will know that they are more likely to be responded to the immunotherapy response. So, but one of the current issue for MSI testing or molecular testing typically relies on two methods. One is the IHC, and the second one is using different molecular assays, which is actually very expensive and at the current moment, and also resource intensive, especially because of the low prevalence of MSI in prostate cancer, which is only about 3%, which actually limited the pre-screening for the mutations. But that's where, on the other hand, we have this routinely collected H&E images at the diagnosis. So what we're trying to think right now is if we can build an AI system or using AI tools to directly infer molecular alterations directly from the H&E images. So just a little bit more background of some of the challenges that we're trying to deal with the H&E whole slide images is many of you already know that this is the large images. Sometimes it can be more than 20 gigabytes per slide. So it's going to be really hard for computers even supercomputers to trying to training a model on that. And on the other hand, for molecular prediction or any other outcome prediction, training on this type of models, usually we don't have enough labeled data for training a large models. So one of the basic simple way to do this is the first step is we're trying to divide that whole slide images into smaller tiles. Then we use some encoders to get some representations of each of the tiles. Then we have the individual tile level predictions for each of the tiles. And those predictions are those labeled wise for each of the tiles are sharing the same label. So which shows that many of you already noticed that one of the tiles is totally different looking from the other tiles, but they're sharing the same label. So there's so much of noise in the supervision here by training in this way. So most recently, there's maybe a better way. It's called multiple instance learning, where instead of doing the aggregation at the prediction level, we're doing the aggregation at the tile presentation level. We're trying to find or weight more on the tiles that is more associated with the molecular alteration and weight less on the tiles that is not associated with the molecular alteration. But there's still a challenge of training a model from scratch, this kind of model from scratch, because when we reduce that tile level prediction into an aggregated slide level representation, we actually reduce the amount of data that we can use for training. So it's still, for any downstream applications, still going to be hard to train a robust performed prediction models. So for the past couple years, there's so many foundation models, just like Bill just mentioned, in large language models field or any other field, and also in digital computational pathology field, there's a lot of foundation models. For the last couple of years, there are maybe every 2 or 3 months there's new published foundation models that we can use. So just like Bill just mentioned, the general idea of foundation models for pathology is also those models are pre-trained on large, massive amount of data. They're trained with self-supervised learning. Usually, they don't need any outcome label to supervise the model. So the general idea of that model is we're trying to grab the general morphology representation we can learn from all the tiles. And usually they're trained from pan-cancer data, a massive amount of data. And with the help of transfer learning, so once we learn those general knowledge, then we can use transfer learning to get a more task-specific representation for each of the downstream tasks that we might be interested. So in our task, we're trying to use foundation model plus transfer learning and trying to do a molecular alteration prediction from the H&E images. So this is just the overview of the framework that we built. So there are so many components here. From pre-processing, we do cancer detection, and we do feature extraction, then we finally do MSI classification there. But the general idea is we actually using transfer learning in a sequential way. We have a sequence of foundation model, and we're training them for each of the tasks. Finally, towards to the MSI prediction from the H&E. So this is a little bit more details of each of the steps. In the first step, we're following the standard pre-processing way. So we, from the input large whole set images, we divide it into tiles, and we do standardized pre-processing, trying to identify the tissue region. So all the other downstream analysis are only focused on the tissue area. Then the second step, where we're trying to do cancer detection. The reason that we want to do cancer detection here as the intermediate step is because that we have the assumption that most of the molecular alteration or MSI mutation are going to happen in the cancer regions. So that's why we also want to kind of restrict our model into looking at the cancer regions. So for this model, we also use the transfer learning with the pre-trained model built by our awesome collaborator from NCI, Stephanie. So a little bit about how we train their model is they actually involved in the human in the loop training. So they have AI predictions that have the pathology to correct the predictions, like doing this in the cycle to fine tune the model. So with the pre-trained model and transfer learning, we are able to fine tune the model to do a really good job on cancer region identification for 35 using only 35 our in-house data set. So I'm going to skip this slides. It's actually just using the foundation model. A lot of them exist. So using those foundation models to get a general representation of the features that we for the tiles. And for the last step, we also used another foundation model, which is pre-trained on a lot of different tasks, about 100 different cancer subtype classification tests, including like cancer grading, disease staging. And so we're trying to see if we can actually fine tune that model, foundation model, to predict MSI prediction. So just a little bit about the data set we use. Our model was trained for using two data set, UW and the TCGA public data set, and also we validate our framework on the clinical trial data set from the UK. So a little bit about the prediction performance that we get. We did internal, external validation. The performance looking good in terms of AUC, and also it looks consistent for different specimen biopsy or surgical samples. And I'm going to skip this slide. So the point is why we even doing like using foundation model and transfer learning to start, but we actually didn't start with that. We started with training the model from scratch for the MSI prediction. And as you can see here, this is a performance by just without using transfer learning. The performance of prediction of MSI from H&E is pretty low, like close to random. So also we can see the highlight part, like a little bit interpretability of how what the model sees in terms of the MSI predictions. But I'm going to skip that, and we're going to introduce the second part, which is awesome work, which is done by our awesome summer intern, Matthias, which is undergraduate student at UW. And he is thinking about how can we make this better or more interpretable, and also by consider the spatial distribution of the morphology of the cells within the H&E, which is actually was not considered in the transfer learning or training from scratch procedure. So he did the clustering and did the spatial feature extraction and used those spatial statistical spatial features to using the machine learning model to do molecular alteration prediction. But This is how it looks on the clustering of the slides. We're looking at spatial map of the clusters. And the sad news is with the spatial consideration, actually for MSI prediction on the internal validation data set, the highest we can get with the 20 number of clusters is actually a lot worse than the foundation model with transfer learning. So we did a little bit more into this and trying to see if it actually going to help for any of the molecular predictions. So we did experiment and it's still preliminary study. We found that actually the spatial information maybe help with some of the molecular predictions, but maybe not helpful for the other ones. But we're going to improve this approach in the future works. So a little bit conclusion, my key takeaway is that we use foundation model, transfer learning is going to help us if we have limited data set. And also, I think it's really important to take into those spatial information or more interpretable features into the model, either improve the prediction and also the performance. So, yep. With that, I'm going to just thank our awesome collaborators and everybody contribute to this project. Thank you.